Very warm welcome to all of you. Today I present my research topic in 41 COVID-19 conference. I'm thankful to organizer to giving a prestigious opportunity to present my research work that's related to phytocompounds in inhibiting the fusion mechanism of COVID-19. My research title is Ellagic acid and Liquiritin phytocompound play pivotal roles in inhibiting the fusion mechanism of newly COVID-19 that inhibit virus entry in host cells. The last 2-3 years we could fighting for the medical health sector. In this time interval, the whole human civilization was facing serious problem, pandemic outbreaks in all over the globe. Due to COVID-19, no medicine was working in this pandemic. In this view of potential threat of pandemic, scientists around the world have been running to understand SARS-CoV-19 and investigate the pathophysiology of this disease to find out the potential therapeutics and invented novel potential drug moiety. In this current situation, we have a research to COVID-19 virus. We have investigated the COVID-19 virus spike protein. That proteins like ATM card, to swap them, then money outbreaks. Therefore, that COVID-19 is a virus-borne disease which is highly virulent virus of the SARS family. The COVID-19 virus assembly have trimeric spike protein. That protein's main tool to the virus, which may possible to virus enter inside the host cell. Therefore, spike protein is the main target to COVID-19 therapeutics approach. Main target is COVID-19 main approach. The spike protein is made up of the two parts. The spike protein is made up of one trimeric helix proteins. They divided in two parts, S1 parts and S2 parts. The S1 parts is receptor binding domain, and second the conformational trimeric regions that help to conformational change and initiate the proteolysis of host membrane and make it the possible to enter the virus via phagocytosis. We have analyzed the S2 region made up of the helix reconfirmed inhibited helix region HR1 and HR2. During the virus entry, S1 region, that's called the receptor binding domain, bind with the host cell receptors, and S2 region HR1 and HR2 make a fusion machinery with the host cell and proteolysis the membrane, enter the virus assembly inside the host cell, and virus enter into the host cells and propagate and make the consequence of COVID-19 virus. Present research, we have the evaluate the virus entry mechanism, we have a target the inhibiting the virus entry through phytochemical compounds. Plants are very useful to making drugs and active pharmacological ingredients. In present research, we have or make a plant to phyto-compounds that inhibit the virus entry into the host cells. Without entry into the host cells, virus doesn't virulent. This is the main phenomena of virus-borne disease. In current research, we have choose two phyto-compounds, that Ellagic is polyphenol found in various vegetables and herbs. Ellagic Liquiritin found in a very much amount of sweet roots is a very common Ayurvedic and Chinese medicine used drug. In present study, we have assessed and investigate the In-Silico study of Phyto-Compounds in COVID-19 spike protein. In present study, we have done the molecular docking study between the virus spike proteins make as a receptors and phytocompound is make as a ligand. For achieving that present study, we have firstly make the phytocompounds with the help of chemistry. They freely downloaded in a ACD labs. First of all, we make the ellagic acid that chemical identity number is 5281853, and liquiritin that chemical identity number is 503737, and one reference molecule that's called V607. This molecule is a known in vitro Mars coronavirus. First of all, we make the 2D structure of ellagic acid, liquiritin and V607 molecule. Afterwards, we download the spike protein of Mars coronavirus. We have downloaded we have downloaded that PDB sites. The ID of spike protein is 2EWS. After the download the receptors and making with the help of chemistry phytocompounds, we have upload that molecule in a docking software that's called the Pyrex 4.1. And after uploading the documents, make the macromolecules and ligands with the help of tool of Pyrex. After the making of PDQT files, we molecular dock that one by one ellagic acid, liquiritin, V07 with Mars spike protein. We have for protein. After the molecular docking with the help of Discovery Studio, we analyze the receptor ligand binding analysis with the help of discovery studios that we assess which amino acid sequence number bind our ligand molecule and how the hydrogen bond, non-covalent bond make the binding very specifically. We discuss about that result. We have docking with the ellagic acid and liquiritin. We have found the very good and very drastically results. First of all, we molecular dock V07 with spike protein and found minus 4.7 kilocalorie per mole binding free energy, and afterwards we docking with ellagic acid liquiritin and sequentially found minus 7.3 kilocalorie per mole and liquiritin minus 9.3 kilocalorie per mole binding energy. The binding energy reflect that our molecule formally bind with the spike protein. Afterwards, we have found the data of binding energy. We assess the protein. We assess the spike protein and ligand binding analysis with the help of discovery studio. We have seen that our ligands bind in a S2 region of spike protein in between the HR1 region and HR2 region. Our ligands bind in a 809 amino acid and 11.03 amino acids. That directly reflect that the spike protein S2 regions, intermediate regions that responsible of proteolysis and initiate the phagocytosis. Our Phyto-Compounds easily inhibit that reaction. Fusion machinery collapsed with the COVID-19 virus with the help of these ligands. So therefore we have very much confirmed that our Phyto-Compounds very helpful to inhibiting the spike protein inhibitors, and reflect that the spike protein inhibits and hinder the phagocytosis reaction of particular virus, coronavirus, that's entry will be hinders. Therefore, we have very confirmed and sure that the virus entry hinder that our molecule, which help for breaking the entry of COVID-19 virus with the help of these ligands. So definitely we have said that the molecule ellagic acid and liquiritin Phyto-Compounds are very much useful for COVID-19 therapeutics agents, and that virus and also those viruses, these mechanisms are similar to COVID-19 virus, also helpful to hinder the entry or inside the host cells. So we have definitely said that the ellagic acid liquiritin to have a broad and potent antiviral function to other viral pathogens like HPV, HIV, HPV, HBV, and HIV, etc. This is my research. Thank you all for